Good morning and welcome to Immunic's IMU-935 webcast. My name is Jessica Brew, Head of Investor Relations and Communications at Immunic. I will also be the moderator on today's call. The purpose of today's webcast is to discuss our positive unblinded safety, tolerability, and pharmacokinetic results from the SAD and MAD parts of our phase 1 clinical trial of IMU-935 in healthy human subjects. In addition, we will discuss newly available preclinical in vivo data for IMU-935. Speaking on today's call are Dr. Daniel Vitt, our Chief Executive Officer and President, Dr. Andreas Müller, our Chief Medical Officer, and Dr. Hella Kohlhoff, our Chief Scientific Officer. Please note, all participants will be in listen-only mode, and this event is being recorded. After today's presentation, there will be an opportunity to ask questions. If you joined the webcast via the Zoom client, there are two ways to submit questions. You can either submit your questions in writing via the Q&A tool of the Zoom portal, or if you would like to speak with us directly, please use the raise hand function of the Zoom portal to queue your question. Before we begin, I would like to remind you that this presentation may contain forward-looking statements. Such statements can be identified by words such as may, will, expect, anticipate, estimate, or words with a similar meaning. And such statements involve a number of risks and uncertainties that could cause Immunic's actual results to differ materially from those discussed here. Please note that these forward-looking statements reflect Immunic's opinions only as of the date of this presentation, and it undertakes no obligation to revise or publicly release the result of any revision to these forward-looking statements in light of new information or future events. Please refer to Immunic's SEC filings for a more detailed description of the risk factors that may affect Immunic's results and these forward-looking statements. I now would like to turn the call over to our CEO and President, Dr. Daniel Vitt, to start with the presentation. Daniel, please go ahead.
Yeah, thank you, Jessica, for the kind introduction, and also warm welcome to today's call from my side. Today, we are excited to share first clinical data from our ongoing clinical phase 1 trial with IMU-935, our oral IL-17 inhibitor. The molecule is a so-called inverse agonist of RORγt, an important nuclear receptor responsible for balancing immune responses. But Immunic has 3 products tested in clinical trials right now. First of all, IMU-838 is our most advanced product, which is in clinical phase 3 testing in patients with relapsing multiple sclerosis and in a phase 2 trial with progressive MS. IMU-838 is also currently tested for activity in patients with ulcerative colitis, a hot area in drug development with very high unmet medical need. We expect to read out results from this study in the second quarter of next year already. Our third product, IMU-856, is aiming to restore the intestinal barrier function with a new and innovative approach. This drug is currently undergoing clinical phase 1 testing. In today's call, my colleague and Chief Medical Officer, Andreas Müller, would like to update you on the beautiful first study results from our phase 1 study of IMU-935. This includes part A, the single dose, and part B, a 14-day dosing of healthy volunteers with IMU-935. We also want to take a very brief look at the currently ongoing part C of the study that is evaluating IMU-935 for 28 days treatment in psoriasis patients. Finally, my colleague and our CSO, Hella Kohlhoff, will then present super interesting new in vivo data on the selectivity of IMU-935, which again underlines the unique profile of IMU-935. IMU-935 is a very interesting new molecule which could be used for a broad variety of chronic inflammatory and autoimmune diseases. IMU-935 is a potent inhibitor of the cytokines IL-17A and IL-17F, and therefore could be a good alternative for IL-17 pathway targeting antibodies, but with oral bioavailability. In particular, IMU-935 could be an excellent way to treat psoriasis, an indication where injectables are widely used in target IL-17 pathway in moderate to severe patients. But of course, the potential use of IMU-935 goes much beyond just psoriasis. IMU-935 could, in principle, be used for a big variety of IL-17 mediated disorders and even for treating specific cancers like CRPC. With this, I would like to hand over to our Chief Medical Officer, Andreas Müller, to share more details on the first two parts of the clinical phase 1 trial.
Thank you, Daniel, and good morning, everybody. So my name is Andreas Müller. I'm the Chief Medical Officer. Let me start the presentation with a quick reminder of the general design of our first in-human phase 1 study of IMU-935. There are 3 parts to the phase 1 trial, and we will report here today the unblinded results from the first 2 parts. Part A was the investigation of single ascending doses of IMU-935, with the highest dose being 400 milligrams in a total of 79 healthy human subjects. Part B was the evaluation of multiple ascending doses for 14 consecutive days, with the highest dose being 300 milligrams daily, and in a total of 15 healthy human subjects. Part C is currently ongoing and includes the evaluation of 28-day dosing in psoriasis patients. All parts of this trial are performed with a double-blinded, placebo-controlled design. Next slide, please. Let's start with the results of Part A, the SAD study. The Part A started with a dose escalation of a pilot formulation of IMU-935 in 5 patients cohorts, which is called Part A in all slides here. The dose escalation was then further continued with a new and improved powder in capsule formulation, which is designated in the slides here as Part A-N. This new formulation was also used in Part B and will also be used for all of the dosing in the ongoing Part C. Next slide, please. This slide summarizes the safety findings in the Part A-N of the Part A. Single ascending doses of IMU-935 were found to be safe and well tolerated. In particular, there were no serious adverse events. There were no dose dependency in adverse events. There was no maximum tolerated dose reached, no trends for post-dose changes in any laboratory parameter. There were no related adverse events in the active treatment group regarding any of the laboratory parameters, and no medically relevant changes in vital signs or 12-lead electrocardiograms as compared to placebo. Next slide, please. This slide presents the most common treatment emergent adverse events found in Part A-N occurring in more than 2 subjects. As you can appreciate from this slide, these are mainly unspecific adverse events observed, such as headache, dizziness, catheter side reaction, constipation, and fatigue. Next slide, please. This slide is more of a deep dive into the treatment emergent adverse events. There were no serious adverse events and no treatment discontinuations in this Part A-N due to AEs that had been observed. There were very few AEs of moderate intensity, and there were mostly events unrelated to study medication and also equally distributed between active treatment and placebo, given the 3-to-1 randomization in this trial. There was 1 severe AE and very few laboratory abnormalities in this trial, but however, all of these events for the active treatment groups can be directly linked to either life events, concomitant medication just prior to the end of the study visit at 14 days after dosing, where most of them were observed. For full disclosure, we provide full information on these events in our extended information section of the slide deck, which will convince you that the nature and the time course of these events cannot be seen as a consequence of study medication. So we really have a very detailed explanation of these events. Additional, these events seem to be incidental findings, given that they're distributed equally between active treatment and placebo in exactly the same ratio as randomized between these treatment arms. In full analysis of all laboratory parameters, there were no trends for any changes over time as compared to placebo control. There were also no adverse events related to vital signs or ECG in this Part A-N. By all accounts, this safety and tolerability profile is quite favorable for a first in human trial. Next slide, please. As an illustration, I would like to show you some selected safety data from Part A-N. The full data set is available in the extended information section of the slide deck. And as an example for hematological parameters, and these are parameters that regularly adversely affected by other immunomodulatory drugs. The left part of the slide shows the time course of the neutrophil count in blood and provides that no changes in the number of neutrophils over time has been observed as compared to placebo. The right side of the slide shows you the shift graphs, showing whether any post-dose values had changed as compared to baseline. A tight distribution of data points in the central quadrant indicates no meaningful changes from baseline to any post-dose time point. Next slide, please. This slide provides the evidence for the lack of changes over time for alanine transaminase, or ALT, one of the most important liver enzymes typically associated with a possible liver injury. So we have this just shows you that there was no change in the ALT over time here in this part A-N. So next slide, please. And finally, this slide provides the Hy's Law chart, which regulators and also clinical developers find very useful to estimate the potential for drug-induced liver injury, which would be indicated by the presence of data point in the highlighted section of concurrent increase of liver enzyme and bilirubin. As you can see, there were no such data points in part A and A-N. These are basically the two parts combined here. Next slide, please. In conclusion, the power and capsule formulation provided favorable pharmaceutical properties during first dosing in humans, and the excellent safety and tolerability profile allowed a very smooth transition into the next and probably more important trial, part of this trial, the part B. Next slide. So we continue the presentation now with the results of part B, the multiple ascending dose part in healthy human subjects. First of all, part B explored two subject cohorts, a lower dose cohort of 150 milligram once daily dosing called QD, and a higher dose cohort of 150 milligrams twice daily dosing called BID. The doses were selected based on the findings from part A to provide dosing alternatives with predicted trough levels well above the expected minimally therapeutic level based on ex vivo evaluations of human lymphocytes. The right side of the slide provides some overview of the structure of the trial, consisting of a 14-day dosing period and a follow-up period. Next slide, please. This slide summarizes the pharmacokinetic properties of the powder and capsule formulation of IMU-935 used in this trial. The observed pharmacokinetic profile lends itself favorably for investigation in further clinical trials, we believe. In particular, IMU-935 has shown a dose linear pharmacokinetic in the dose range examined. The plasma half-life of IMU-935 is close to 30 hours based on a non-compartmental analysis. Also, once or twice daily dosing results in very fast steady state trough levels to be achieved within first week of dosing, and the steady state trough levels are stable over time. The accumulation factors for multiple dosing were found to be in a range that should result in reproducible trough level distribution in a larger patient population. We believe that's also important for future clinical trials. The time course of average IMU-935 plasma levels, or mean plasma levels, on the left also contains the IC50 and IC90 values for inhibiting IL-17F release. IL-17A is quite similar. It's just as an example here, the IL-17F release from human lymphocytes in previous ex vivo studies. This indicates that these two doses used in part B produce drug levels that should already be well within the expected therapeutic window of IMU-935. It is to be noted that these two dose levels here from part B were also used in part C, or will be used in part C, for 28-day dosing in psoriasis patients. Next slide, please. This slide summarizes the safety findings in part B of this trial. Multiple ascending doses of IMU-935 over 14 days were found to be safe and well tolerated. In particular, there were no serious adverse events. There were no dose dependency adverse events. No maximum tolerated dose was reached. There were no trends for post-dose changes in any lab parameter. There were no adverse events regarding any lab parameter, and there were no medically relevant changes in vital signs or 12-lead ECGs as compared to placebo. Next slide, please. This slide summarizes the overall incidence of treatment emergent adverse events in Part B. There was no dose-dependent increase in adverse events, and the incidence of adverse events was well distributed between active treatment and placebo. Next slide, please. This slide again presents the most common treatment emergent adverse events found in Part B that occurred in more than one subject. As you can appreciate again, that these are mainly unspecific adverse events observed, such as headache, back pain, epistaxis, constipation, and catheter site reaction or irritation. Next slide, please. This again is a more deep dive into the treatment emergent side effect profile, adverse event profile. There were no serious adverse events and no treatment discontinuations due to adverse events have been observed. All adverse events were mild in intensity, except for one each in active treatment and placebo. The moderate event in the IMU-935 arms was a catheter site reaction not related to study medication, in particular considering, of course, that IMU-935 is dosed orally. There was no AE of severe intensity in Part B. In full analysis of lab parameters, there were no trends for any changes over time as compared to placebo. Event based on lab abnormalities or on vital signs observed in Part B. There was a mild PR interval prolongation seen in a subject dosed with IMU-935 during ECG evaluation. PR prolongation is a known benign finding that can be observed in healthy young individuals. It is known to be of no prognostic or therapeutic consequence. However, for full disclosure about this event, there's a lot more details given again in the extended information section of this slide deck. It also needs to be noted that not a single QT interval prolongation in ECG was found in any subject at any time point in Part A or B exceeding 440 milliseconds. By all accounts, this safety and tolerability profile is as good as you can obtain in a thoroughly monitored multiple dosing trial as part of a first-in-human investigation. So we're very excited about these results. Next slide, please. Similar to what I've shown for the SAD part earlier, I would like to present some examples for lab values again. This slide shows the time course and provides that no changes in the number of neutrophils over time has been seen as compared to placebo. Additional shift tables indicate no change outside the normal range at any observed time point. Next slide. This slide again showing that there's no evidence for increase over time for alanine transaminase or ALT, one of the most important liver enzymes typically associated with a possible liver injury as compared to placebo. All of the other liver enzymes data and bilirubin are also contained in the extended treatment, extended information section here. Next slide. And finally, again, this is the Hy's law chart for Part B. This slide showing no occurrence of concurrent increase of liver enzymes and bilirubin. The data on IMU-935 and placebo also plot right on top of each other here with no values in the Hy's law quadrant highlighted. Next slide. In conclusion, the favorable safety, tolerability, and pharmacokinetic properties for IMU-935 in daily dosing in healthy human subject allowed a very smooth transition into the next part of the trial. This is the Part C. The Part C, of course, with the 28 days dosing in psoriasis patients currently ongoing. We already made the announcement earlier that the recruitment had started. So next slide, please. So let me give you a quick overview of the ongoing Part C. Part C will be a double-blind, placebo-controlled study with 28-day dosing in psoriasis patients with moderate to severe disease. 2 separate subject cohorts are planned to be investigated using the same dosing regimen evaluated in Part B. All in all, approximately 52 psoriasis patients are planned to be included in Part C. Next slide. The lower dose cohort of 150 milligrams IMU-935 once daily is already ongoing and actively recruiting further patients. Once this initial cohort is fully recruited, the study will immediately continue the recruitment of the second patient cohort using the higher dose of 150 milligram IMU-935 twice daily. The right side of the slide shows the general design of the dosing and observation periods in Part C. The study will evaluate the safety, tolerability, as well as the trough levels in these psoriasis patients. However, we will also evaluate pharmacodynamic markers, as well as we will make an ongoing evaluation of skin parameters, including PASI score. Before I hand over now to my colleague Hella, our Chief Scientific Officer, who will give you some very exciting updates on new preclinical and tox data, I would like to really thank the study team of Immunic involved here, Irina, Maria, and Frank, for wonderful work done, and really would like to thank them for their work. So over to Hella. Thank you.
Thank you, Andreas. So now we have seen that IMU-935 exhibits extremely good safety and PK properties in healthy subjects. And I'm absolutely convinced that soon we will see efficacy in psoriasis patients as well. The study has started and we just have to be a bit patient for some more months. But what I can show you now is the outstanding selectivity of IMU-935, because to our knowledge, it is the first ROGAMA-T inverse agonist that can modulate ROGAMA-T in a cell status specific manner. Please let me remind you, ROGAMA-T is the main transcription factor for the differentiation of Th17 cells and the expression of IL-17A and IL-17F. To treat autoimmune diseases, the inhibition of Th17 cell differentiation and IL-17 production is key, and IMU-935 is extremely potent here. But there is another physiological role of ROGAMA-T, and this is the maturation of T cells in the thymus, the so-called thymocytes. This thymocyte maturation is necessary to produce a broad repertoire of T cells to support the immune system to fight external challenges. In addition to an impaired T cell repertoire, an inhibition of this process may lead to lymphoma development. Therefore, the goal for IMU-935 development was that 935 can potently inhibit Th17 differentiation and IL-17 production, but allows normal thymocyte maturation. And indeed, we think that we've been successful here because IMU-935 does not affect thymocyte maturation in our experiments. And all these data I will show you now on thymocyte maturation were performed by our wonderful collaboration partners, Professor Su-Ming Sun and Hong-Min Wu from the City of Hope in Duarte. Next slide, please. First, I want to show you the in vitro data. For comparison, we have used the small molecule ROGAMA-T inhibitors compound 1 and MRL-871, as they have been published in peer-reviewed journals to inhibit Th17 cell differentiation and also to inhibit thymocyte maturation, what is reflected by a loss of cell viability of thymocytes. On the left, here you see the differentiation inhibition of murine Th17 cells, which were treated with 500 nanomolar of the respective inhibitor or sintirogon, an agonist, so an activator of ROGAMA-T. You clearly can see that all inhibitors and the ROGAMA-T knockout inhibit Th17 differentiation as compared to DMSO control, whereas the activator slightly induces Th17 cells. On the right, we investigated the impact of all compounds on thymocyte maturation and used viability as a readout here. The agonist sintirogon induces viability of the cells, as ROGAMA-T provides a survival signal during thymocyte maturation. In line with this, the ROGAMA-T knockout, closed down, strongly inhibits the viability of thymocytes. When it comes to IMU-935, you clearly can see that it does not reduce the viability of thymocytes and behaves like the vehicle. Whereas the inhibitors MRL-871 and compound 1 strongly inhibit the viability of thymocytes. Next slide, please. And now we can ask, can this be translated into the in vivo situation? And yes, it can. In this acute mouse model, we treated animals for 72 hours with vehicle, IMU-935, the inhibitor MRL-871, and the agonist Sintivorgon. And already after 3 days of treatment, you see a strong reduction of the thymus size with a positive control MRL-871 and of the number of cells in the thymus. Whereas IMU-935 does not impact the size of the thymus or the cell number in the thymus. Next slide, please. So to further corroborate these findings, we performed the chronic mouse model as well. Here we used a twice-daily treatment for 28 days. What immediately stands out is that, again, the positive control, in this case, compound 1, significantly reduces thymus size and cell numbers, while IMU-935 behaves like the vehicle control and the activator and does not impact the size or the number of cells in the thymus. In addition, IMU-935 does not impact the thymocyte maturation, what is not shown here for time limitations. Next slide, please. So combined with the previously shown data on the gene regulation by IMU-935 during Th17 cell differentiation and thymocyte maturation from July of this year, we believe to have a clinical stage molecule in hands that has all the desired effects on Th17 differentiation and IL-17 production, but omits the undesirable effects on thymocytes. This unique and cell status selectivity on raw gamma T and lack of off-targets is further confirmed by the preclinical safety and toxicity studies we have conducted in rats and dogs. We have conducted the 28 days and the 3-month safety studies and will perform the long-term studies for 6 and 9 months during 2022. The major findings on these studies were that the NOAL from the 28-day toxicity study was maintained in the 3-month study, meaning that a longer treatment duration with IMU-935 does not change the safety profile. Due to the target ROGAMA T and the issues known from other molecules, thymus and liver were the main targets of interest. Again, no findings were reported on the thymus or liver enzymes in these studies. And in summary, we believe that these data from the preclinical safety studies and the in vivo models for thymocyte maturation paved the way for IMU-935 to be a best-in-class oral IL-17 inhibitor. Back to Daniel.
Yeah, thank you, Hella, for the wonderful data. Quite exciting. And I have now the pleasure to summarize that once again here. So IMU-935 showed a very favorable safety, tolerability, and PK profile in this phase 1 clinical trial with no serious adverse events seen in the SAD and MAD parts. In particular, IMU-935 was safe and well tolerated in the 14-day repeated oral dosing in healthy volunteers at doses expected to clearly exceed required therapeutic dosing. The molecule's outstanding selectivity profile on Th17 over thymocyte development was confirmed in an impressive fashion in acute and chronic mouse models. The drug is currently being tested in psoriasis patients with initial data expected in the 2nd quarter of next year, setting the stage for a potential best-in-class oral psoriasis therapy. And IMU-935 may offer extensive potential beyond psoriasis in other autoimmune diseases. But of course, we have also a lot of other things ongoing, and we expect next year to be Immunic's year, with a lot of important milestones and value inflection points ahead. One of the most important milestones for Immunic are phase 2 top-line data from the ulcerative colitis phase 2 trial with IMU-838, which we expect to be available in the 2nd quarter of next year. As already mentioned earlier today, another important milestone next year comes from IMU-935 studies. We expect initial safety, efficacy, and efficacy data from the 4-week Part C in psoriasis patients as part of the phase 1 study, also in the second quarter of next year. And on top of that, we recently started a phase 1b trial in patients with CRPC. First data from this study is expected to be available in the third quarter of 2022. For our third molecule, IMU-856, unblinded SAD/MAD safety data in healthy volunteers is expected to be available in the third quarter of next year. Yeah, with that, we can, I think, enter the Q&A session.
Yes, thank you, Daniel. Also, thank you, Hella and Andreas, for walking us through the clinical phase 1 SAD and the new preclinical data for IMU-935. As Daniel mentioned, we will now begin the question and answer session. As a reminder, if you joined the webcast via the Zoom client, there are 2 ways to submit questions. You can either submit your questions in writing via the Q&A tool of the Zoom portal, or if you would like to speak with us directly, please use the raise hand function of the Zoom portal to queue your question. At this time, we will pause momentarily to assemble our roster.
Dann sieht man die.
Our first question comes from Yasmin Rahimi and Jesse Foe at Piper Sandler. Jesse, please unmute yourself and go ahead. Jesse, I see you unmuted, but we can't hear you.
Can you hear me?
Yes. Hi, Jesse.
Okay. Hi team. Congrats on the data and thank you for taking my questions. I had 2 of them, if that's okay to ask.
Sure.
Okay. My first one is, could you please highlight what depth of the cytokine and inflammatory biomarker data was collected in the MAD portion? And were there any dose-dependent differences? And how would these biomarkers establish efficacy in psoriasis?
Okay, so as the MAD clinical study was performed in healthy volunteers, we did not take samples to determine IL-17 or other biomarkers because in human beings, in healthy human beings, the levels of the cytokines are so low or more or less undetectable. What we have done is that we earlier performed ex vivo data where we used PBMCs, human PBMCs from healthy volunteers, and they were stimulated ex vivo and at the same time treated with IMU-935. And actually there we see this dose-dependent inhibition of IL-17A and IL-17F.
Great, thank you. And then my second question is that we understand that the 150 mg QD and BID is safe in the Part B, but could you please walk us through what steps went through into selecting these doses for Part C?
So this is Andreas again, and thanks for the question. I think even in Part B, we selected these 2 dosing regimen because we believe that given the results that we had from Part A, in terms of what blood concentrations or what plasma concentrations you had of IMU-935 after 24 hours, which gives you basically an indication of trough levels and expected accumulation factors, that these 2 dosing regimen would be well above the minimally therapeutically drug levels. And that's why we selected them already for Part B, with the expectation, if we don't see any safety signals, which of course we expected not to see, but that we continue these 2 dosing regimen also for Part C. So this was already, I think, a decision done in the transition from Part A to Part B, and now we're continuing these 2 dosing regimen also in Part C, and we very strongly believe that we will see with both of these dosing regimen activity in terms of PASI score and also pharmacodynamic markers in this Part C.
Great, thank you so much. those are my questions. Thank you.
Thank you.
Thank you, Jesse. Our next question comes from Tom Smith at SVB Leerink. Tom, please unmute yourself and go ahead. Tom, you need to unmute yourself, please.
Hi, everyone. This is Mike Cracky on for Tom. Thanks for taking
Hi.
our questions. Can you help characterize the dizziness seen in parts A to N? And is there any reason to expect, any CNS issues moving forward? And do you know if IMU-935 crosses the blood-brain barrier? And then I have a separate follow-up question.
So maybe I start with the dizziness question and let Hella answer about the ability to cross the blood-brain barrier. So these are unspecific adverse events that we have seen in both part A and part B, and they are a little bit different between both parts because they're unspecific. It's sometimes they happen in such setting when you don't have any specific side effects that are attributable to the drug in itself or to the study medication. We don't think this is a specific issue here because also I think these side effects often are also a cohort phenomenon. Yes, these patients are together in a phase 1 unit, and sometimes a certain cohort has these events and the next cohort doesn't have them. That's the nature of these unspecific adverse events that you see in a phase 1 trial, especially if these healthy human subjects are housed together. And since we could not identify the same unspecific issues again in the part B, for example, I think it has very little prognostic value for the safety of IMU-935. And I let Hella answer the question about blood-brain barrier.
Yes, happy to do so. So actually, we have performed a tissue distribution study with radio-labeled IMU-935, and that was tested in a so-called QWBA study. And actually, we see a normal tissue distribution, so no specific blood-brain barrier or high exposure in the brain.
Got it. That's very helpful. Thanks. And how would you characterize the psoriasis patients that you're going to be looking at in part C of the study? Are these patients that are going to have failed other lines of therapy, or are you restricting to treatment naive patients?
Yes, thank you. Very good question. We haven't specifically published for competitive reasons the specific inclusion criteria for part C. But I can assure you, we expect to enroll patients with moderate to severe disease. So the PASI scores and other skin scores that we allow to go in basically make them moderate to severe disease. And we also require that they failed at least topical treatment. So these are not necessarily treatment naive. So this would be very much in line with a patient population that you would expect for an oral drug in psoriasis, that these patients have to have failed at least topical therapies, or at least one topical therapy.
Understood. Thanks again for taking our questions.
Thank you, Mike. Our next question comes from Ram Selvaraju and Muzamil Salim at H.C. Wainwright. Muzamil, please unmute yourself and go ahead.
Hi, good morning. Thanks for taking our questions and congrats for the data release. So just 3 from us, if I may. Just linking the preclinical thymocyte maturation data to the human safety data, how significant and differentiating do you think this is? And are we already able to see evidence that from a clinical standpoint, this has been validated through the safety data? If not, what else would be needed?
But I think we did all the efforts here exactly to demonstrate that there is no effect. And I think just from a medical scientific point of view, with the in vitro data and the animal data, that's a perfect package to really underline that there is no effect. We just did that because others have seen issues with their molecules, which is difficult to address for ourselves. And therefore, we see that as a solved thing. Secondly, the 3 months tox performed was really, I think, done that early in this development to underline that there's nothing there. And other companies we know have seen, if they had some effects, they have seen that. As far as we are aware, already in the 3 months tox studies, and even earlier, as you see, already in the acute model, you see an effect after 3 days, of course, with a quite intense dosing. But I think that underlines that our drug is expected to be totally free of that risk.
And I would like to add maybe that the thymus, of course, is responsible for maturing lymphocytes, and as you could see from the preclinical data, if you block basically the maturation of these lymphocytes in the thymus, especially T lymphocytes, that's why they're called T for thymus, you get lower numbers of lymphocytes, which we haven't seen. So I'm not sure this is a complete proof for the clinical yet, but because we dosed only for 13 days, but I think it's a very good indication. That's why I wanted to show you also the hematological data, and the extended information section has basically all of the hematological data that shows that there's really no change in any of the blood cell components in the MAD part. I think at the end of the day, I think it's also a chronic issue where you probably have a risk of thymomas probably down the road if you block the thymus function. But this is probably a much more chronic issue that you can't address in this phase 1 trial.
Appreciate the clarity there. In terms of chronic dosing, how long will it take for the necessary safety data to be available in order to enable IMU-935 clinical assessment? Would it be 12 weeks or 24 weeks in terms of chronic dosing?
So I think at the moment, with the availability of 3 months tox data, we would be able to basically dose for 3 months, and that's kind of the regulatory framework. We will start the longer tox studies in early 22, which would allow us to dose longer. But the part C, I think we are based on previous data, not only from Vitae Pharmaceuticals, but also looking at the phase 2 studies of all of the drugs that went through clinical development in phase 2 and then on to phase 3. It shows that 28 days is enough time to show a half maximum effect that you would probably get in a phase 2 trial. So that's why we believe that 28-day dosing which was available at that time when we started part C, is ultimately enough to show proof of activity in the part C.
Okay, great. Makes sense. And then just finally, what's the anticipated speed of onset of therapeutic action in psoriasis, would you say?
I'm sorry, tell me a little bit about what the expected what? I may have missed it.
How quickly you think there will be a therapeutic action in psoriasis?
Yes. Yeah, no, I think this is what I just mentioned. We actually had looked at basically several of the phase 2 trials for basically approved drugs, and how quickly the change in PASI and in other scores and in skin activity you had seen in these phase 2 trials, because the phase 2 trials usually are 12-week studies for the primary endpoint, so about 3 months. And you could see definitely that you had kind of a exponential curve with the change in PASI and other skin scores being much higher at the beginning of the treatment, so that already at week 4 you had a half maximum response that you would see at 12 weeks. So that's why, and also we know from the Vitae pharmaceutical data that 28 days was enough time to really show a statistically significant change in PASI score as compared to placebo control. So we believe that being above the minimally necessary concentrations to show a treatment effect based on the IC90 data that we have from ex vivo investigations in human lymphocytes, that we are very confident that we can see good activity in 28 days and basically show proof of concept here in the part C. That was the whole I think rationale for designing the part C the way we did.
Okay, makes sense. Thanks for the clarity and thanks for taking our questions.
Thank you, Muzamil. Just as a reminder, if you have a question, please use the raise hand function of the Zoom portal or use the Q&A tool. Next question comes from Matt Kaplan at Ladenburg Thalmann. Matt, please unmute yourself and go ahead.
Hi, good morning. Can you hear me?
Yes, good morning.
Great. Thanks for taking the questions. I guess first, just wanted to get an idea in terms of where you are with formulating 935. And with respect to, I guess, given the strong PK profile you've seen now with the powder and capsule, what are your thoughts on, is this the final formulation or are you going to work on additional formulations?
Maybe directly answering that. Thank you, Matt, for the question. I think we made a big step here from our pilot formulation to the powder and capsule formulation, which is fine for now and for phase 2. On the long run, for market quality, maybe also considering a tablet, but we have enough time and use capsules so far. I thought it's a pretty good formulation from our point of view.
Okay, great. And then question in terms of how well do you think the IC data ex vivo convert to the minimally clinically effective dose levels that you expect to have in the clinical trials?
I think this is Matt. This is, I think, a very good model because the drug target really is the circulating lymphocyte. Yes, this is where we need to be and where we need to show where we need to have the raw gamma inhibition. So I think that's why we did the ex vivo studies, because I think this is more or less the target organ for the drug effect here.
Okay, great. Well, thanks for taking the questions and congrats on the results.
Thanks, Matt.
Thank you, Matt.
Thank you. Next set of question comes from Govind Singh at JMP, and I received those via the chat, so I will read them. 3 questions around IMU-935 versus Vitae's compound. First, previously, Vitae correlated PK to psoriasis efficacy. Do you think there is something to that analysis? And if so, how might your data look in that analysis?
To be quite honest, I think that there were also a few, I think, problems with the PK profile in the Vitae trial. So I'm not sure how to directly translate the PK from Vitae to our drug. And the reason is that they had seen some non-linear effects in pharmacokinetics and also some changes in from what I understand in terms of steady state trough levels over time. So we have seen a dose linear pharmacokinetic, as well as we have seen very stable trough levels over time, indicating that there's no emerging issue in terms of elimination or metabolization. And so it's, often, of course, you have the question, don't you need kind of a stable drug level on target to have a consistent effect? And with the Vitae data having all these problems in PK, I think it's a little bit difficult to translate this to a drug like IMU-935, where you have a very simple pharmacokinetic with a dose linear stable pharmacokinetic. So I kind of struggle a little bit to make a one-to-one correlation between Vitae and our drug for that reason.
All right, then we move on with his second question. Any way to compare your PK data to Vitae's? It seems like you are able to achieve greater serum concentrations using lower IMU-935 doses relatively and relative to IC90 for IL-17F.
Maybe I take that question. I think, and maybe you need to repeat that. we have in the graphs in the presentation, we have these green dotted lines to make it easier to understand what is the difference between the exposure we have seen in the trial and what efficacy what activity was seen in vitro with IC90 concentrations in the single-digit nanomolar range. So that's so much covered that I think it's something where we believe that both doses are clearly above that level, and the rest we will see going further through the part C of the phase 1 trial.
Okay, and Gobin's third question, any preclinical thymocyte maturation results with Vitae's compound earlier? And if that compound did impact thymocyte maturation, I can't imagine regulatory authorities not caring about this. So what am I missing?
Yeah, so that's more a little bit of a political question here. I think we can't argue for other companies and what they did and others are doing. We have the data. I think it's wonderful that our drug really behaves as it should behave to be a safe and selective molecule. And others may have some challenges there. So Hella nicely showed data from literature as well. So the MRL 871 was published. There's data published in other molecules, which is a reference. And we have shown some of the data here in our presentations. And I think it gives a very crystal clear message here. And we have the data we need for discussions with the regulators.
All right, so that were Gobin's questions. Thank you, Gobin. Next questions come from Andreas Argyrides at Wedbush Pacro. Andreas, please unmute yourself and go ahead.
All right, good morning, and congrats on these early results. Just a quick one on the dosing and how you're looking at Part C to inform whether you go ahead, I guess, in a phase 2 with QD or BID. Thanks.
So thanks, Andreas. I think to a certain extent, the Part C is also meant to give us some indication of dose response and where we are in this dose response curve for IMU-935 on psoriasis for the planning of the phase 2 trial. We would expect, of course, that a phase 2 trial in psoriasis still has several active doses that we need to include, as well as placebo. I think that's the standard design of a phase 2 trial in psoriasis. What we have seen with other oral drugs in their phase 2, if you look at 28-day data, that you get somewhere a PASI score change of about 25 to 30% as compared a decrease from baseline, and in placebo you really you're still at 10% or less. And if you see that that same behavior here in the Part C, where our 935 arms reach that level, I think it indicates that this is kind of the expected activity level that we and guide our dose selection for the phase 2. But we, of course, we're designing already the phase 2 study. We're preparing for the submission, but in a lot of ways, I think these data will inform a little bit where we are on the dose response curve. I hope that's answering directly your questions.
It is. Thank you. Appreciate it. And good luck on the progress. Thank you.
Thank you, Andreas. Last set of questions I received from Zach Bejala at Roth Capital. Are there expected to be any unique AEs that psoriasis patients could have related to healthy volunteers, or any other consideration as we think about translating these results to psoriasis patients?
So I think at this time, and based on the Part A and B results, also I think we wrote this specifically in the press release, we haven't found a specific adverse event or set of adverse events that are specific or seems to be specific for IMU-935. So in that respect, we have not yet a set of AEs or or lab abnormalities that we should look for in the Part C or an EDDI investigation of psoriasis patients. Psoriasis patients, of course, they have their own set of comorbidities sometimes, and I think the most important comorbidity is what's called metabolic syndrome, which I think includes something like high blood pressure or a propensity to have increased liver enzymes already at baseline, for example. So I think this is definitely, I think, when you look in psoriasis patients, areas where they're different from healthy human subjects, and that's where we continue to have very close attention in terms of our monitoring. However, I think we have not seen any signal or any indication that liver enzymes, hematological parameters, or blood pressure is affected by IMU-935. So this is something that we need to monitor in specifically more in psoriasis patients. But in general, I believe that the Part A and B results, I think, is a very good start to showing that there's a very favorable safety profile for IMU-935.
Thank you. Second question from Segba. Just a quick clarification, is IMU-935 a pure raw gamma T inverse agonist, or is there some activity also on DHODH?
So actually, it's a very potent raw gamma T inverse agonist, but we see at 10-fold higher doses, we see inhibition of DHODH as well, but it's much more potent on raw gamma T.
Thank you. And third question from Segba, and last question for our call today. Not related to psoriasis, but was just curious as it relates to the prostate cancer effort, since it is a slight deviation from your current pipeline indication. Is it possible that you might consider partnering the prostate cancer program earlier than your other programs if the data is positive?
Well, I think here I need to repeat what we always say. We know that we have 3 great molecules in development at several trials. And the company Immunic is really focusing on good interactions with pharmaceutical companies and really getting people up to speed where we stand and what great data we have. And we will be open-minded to talk with companies if such a request comes to us, and we will consider that. But there's no strategy to focus on one or the other. I think it's really having good data, good discussions, and then do the best for the company.
All right. So thank you very much, everybody, for the great questions and the good discussion. This concludes our question and answer session. I would like to turn the call back over to Daniel for any closing remarks.
Yeah, thank you, Jessica. With that, I would like to end today's webcast, and thank you very much for your participation and the good questions we got here on the call. Our team is very excited about the progress of this program, but also of the other programs. And we are looking forward to a super interesting year 22. And with that, take care and goodbye.
Thank you. The conference has now concluded. You may now disconnect.